Welcome, everyone. Thank you for coming out tonight. I want to welcome you to our last episode of BioFrontiers Presents this semester. And I just want to give for those of you who don't know me, my name is Roy Parker. I'm the director of BioFrontiers here, and I work with Leslie Leinwan, our chief scientific officer, to put this whole thing together. And we're helped by terrific staff, including Matt back here, and Kristen, and lots of other people who make this all work. And what I want to do for those who, just as by way of introduction, for those of you who don't know what BioFrontiers is, this is an institute here at CU Boulder that brings together people from very different disciplines, from physics, from computer science, from biochemistry, from engineering, from hardcore biologists, and even ecologists, to try to solve important biological questions. And the idea here is that if you get people together who have very different perspectives and skills, you can advance and make things happen that wouldn't otherwise. And our work really focuses in these 5 areas, things like regenerative medicine. How do we build new biomaterials to help heal our bodies? We have a program on Down syndrome, really because of some very nice philanthropy from the C family. Computational biology. One of the big problems in biology today is how do we use all the data we generate to figure out what it's telling us. Degenerative diseases, this is a passion of many of us. As we all get older, how do we deal with muscular and neurodegenerative diseases? And then also parasites and pathogens, which we all have become familiar with in the last 4 years, but have an even broader impact that we work on and try to study. One of the things that we decided to do, we wanted to do this a few years ago, but COVID kind of derailed our hopes, was to have a series of talks from people in BioFrontiers about what they're doing and about why it's of interest and how it connects to all of you. And we've been doing this to reach out to the community. One of the things that I think we're passionate about here is that as scientists, we have an important role to not only do science, but also to communicate that to the public and try to help the public understand what we do as scientists and why it's of value. And so we've had a series of talks. For those of you who weren't here, we had a talk on heart health from Leslie, a talk on RNA biology from Tom Check, and a talk on emerging viral threats from Sarah Sawyer. Those are all online on our YouTube channel. If you're interested, they're all terrific talks. Tonight, I want to introduce our speaker, Sammy Ramsey. Sammy is a unique hire for our institute. We've never had a physiologist, not really a physiologist, what would I call you? An entomologist, right? Or really an ecologist before. But one of the things that we also believe is that, science should not have boundaries. It should bring people together from different perspectives. And as you're going to hear, some of the stuff Sammy does trying to understand insects can really be impacted by some of the molecular and biochemical kinds of things that we do in this institute. And he helps teach us about the importance of insects in the world. A few things about Sammy. Sammy's really been here 2 years, so he's just getting going. He's really an innovator and leading researcher in how we should understand bees in the world, and particularly how we should understand bee parasites, which, as you're going to hear, is a really important issue. He's also an incredibly and well-known scientific communicator, and one of the classes he teaches here is how to be an effective communicator. He's won many awards, including he's a National Geographic Wayfinder and an Explorer Club award winner, and he's done a lot of terrific things, and we have lots of excitement about having him here as part of our community. And so without further ado, please join me in welcoming Sammy.
Okay. All right, everyone. I am absolutely delighted to be here and so grateful that all of you are here to listen to me prattle on about one of my favorite subjects in the world. I love insects, and it is very hard for me to find any bug on this planet that I do not think of as the most interesting thing that I could possibly have stepped on at some point. And so, at this point, I want you guys to take a moment and just go on a bit of a journey with me. all right? I want you to imagine a world in which a new disease has rapidly spread from one part of the world all around the world. And this disease, while it swept across the world, has killed a vulnerable portion of the populace while forcing others into intense isolation, breaking the normal structuring of the social dynamic woven between them. Yeah, sound familiar? I'm sure you're all well aware that I am talking about the diseases that have heavily impacted honeybees over the past few decades. Why? What were you thinking of? There is something called a pollinator pandemic, and we have been well entrenched in the concept of pandemics and the impact that they cause on human populations, but we give very little thought to the fact that the same thing happens in the world of pollinators. And there was a time for which I was trying to get a lot of other researchers and the individuals in charge of grant funding to understand that this is a huge, huge issue, and one that we can actually get in front of if we spend our time focusing on these issues before they actually arrive at our doorstep. Well, it wasn't until I started calling it a pollinator pandemic in 2020 that people really started listening, because we had just the right set of circumstances to make that very, very real to us. Now, consider the pandemic that we just went through forced people into isolation, and the virus was killing about 1% of the population. With honeybees, 33 to 51% of our honeybee population is lost every year. In the middle of this pandemic, now this is the set of circumstances where I would expect if we were going through this, we wouldn't have a tough time getting people to wear masks. Imagine if COVID was taking out a third to half of the populace. Now, these insects have been dealing with it and dealing with it in stride. But they cannot do this forever. There's an unsustainable level of losses every year with our honeybees, and it is finally reaching that tipping point where we're very concerned. Because here's what happens. Each year, we lose a substantial proportion of our pollinators, but we have a trick up our sleeves. Beekeepers have the capacity to take whatever colonies are left and split them in half. You add a new queen to that second colony that you've established, and you've recouped your losses. Except if we start reaching the point where our average losses each year are above 50%, and that's what we're trending toward. So my research is focused on keeping us from getting to that point. Now, why does all of this matter? Well, I would just like to throw it out here. You would not enjoy your lives as much without the honeybees. There's an interesting quote that's been going around the internet that if we were to lose our honeybee population, that human beings would die within 5 or 10 years, something like that. Have any of you come across this quote? It's attributed to Albert Einstein, who was a physicist. I don't know why he would be talking about insects, but people love attributing things to Albert Einstein when they want to sound smart. This was not a quote from Albert Einstein. There was no set of circumstances in which he said this. This was a quote from someone whose first name was Einstein, and we've just run with it ever since. But it is, however, inaccurate. Populations have survived for substantial periods of time on wind-pollinated crops such as corn and rice. But imagine if your diet was pretty much all one color. Imagine if you lost 2/3 of the fruits and vegetables available to you. I'd like to just name 3 that I think we enjoy quite a bit. Coffee. Anybody a fan of coffee? Yeah, okay. Avocados? We down with guacamole? Yeah, all right. And then, I don't know, apples, apple pie. But like, the fact that we would have to do without things like coffee is already a huge deal. Consider this, though, that $15 to $18 billion of the United States economy every year is wrapped up in the pollination services of this one insect. Consider further that global food security is based on the capacity of these insects around the world to pollinate crops at a rate that is far more efficient than any of their competitors. This is the reason why people keep pushing for the whole save the bees thing. It's not because honeybees produce honey. That's a very small part of what goes into the economy and what makes them so valuable. They are actually the 3rd most valuable livestock in the world, just after cows and pigs They're even more valuable than chickens. Now, what makes them so incredible at what they do? I mean, honeybees aren't even native to the United States. This is the European honeybee that we're talking about. They were brought over from Europe along with the settlers. There were honeybee skeps on the Mayflower, which is pretty incredible. And when they arrived, the Native Americans referred to them as the white man's fly. Stay far away from the white man's fly. It will hurt you. Fascinating, but why do we focus so much on these? Well, their nearest competitors, other superorganisms, and when we talk about superorganisms, we mean a creature that is composed of a whole bunch of creatures all together, all working together towards the same goal. Superorganisms are incredibly efficient creatures. But the ones that we have here in the US, their population sizes range from a few hundred to maybe 1,000 individuals, while a colony of honeybees has 20 to 60,000 workers. And so their ability to fly to all of these different flowers, move pollen back and forth, and effectively pollinate them, allowing them to produce the fruits and vegetables that we consume on a regular basis, they are going to be incredibly efficient at that task. They're also generalists, so they're capable of pollinating quite a wide variety of crops. 67 of the 105 crop commodities that we rely on this planet are pollinated by these bees. So as superorganisms, we have to remember that a single bee is not a bee on its own. That bee cannot live by itself. That bee cannot accomplish the wonderful things that a colony of honeybees accomplishes. It's them all together that forms the incredible complex that we are so excited about. And there are a lot of parallels to an actual creature. When they build a colony, they build it out of wax. They extrude wax from between their abdominal segments, and they build their house out of it. Now imagine how much housing prices would go down around here if we could build a home out of something we squeeze from between our abs. Do you know how often I would be in the gym? Incredible stuff. But we can't do that. But the bees themselves can, and they construct this home, and the wax itself forms something like the skin of the colony. It forms the bone structure of the colony. It also forms these cells that allow them to store incredible sources of food. They collect pollen, they squeeze it together and add some secretions and enzymes from their digestive tract to it and create something called bee bread, an incredibly nutritious bundle that they're able to consume and use to create the food that they regurgitate into the cells of their offspring. Pretty cool stuff. But it gets cooler than that, because where things really get fascinating is the honey. Honey is a solution that is so hypertonic that bacteria can't persist in it. As a result, it doesn't spoil. We found honey in King Tut's tomb that was still edible. It's an incredible, incredible resource because you can keep it around as long as you want, and it is a remarkable source of energy to the entire colony. So, you might have guessed that there are a few creatures that want in on this whole situation. And for good reason. I mean, while the honey is incredible, while the bee bread is incredible, you still have not scratched the surface of what makes them so remarkable. Now, normally, the way things work ecologically is that creatures have to opt into one of two styles of producing offspring and going about their life cycle. One style of producing your offspring has organisms investing very heavily in 1 or 2 or maybe 3 offspring, but they invest all of their energy and resources into them, and as a result, their mortality rates are very low. These organisms typically survive very, very well. But for most insects, the way that they do things is they lay thousands upon thousands of eggs and never even live to see them hatch. Most of the time, they die before their offspring are even born. They invest very, very, very little energy in each one. And that's a system that has its merits as well, because it's more of a numbers game. If I lay enough eggs, even if mortality is 95%, which is the typical mortality for creatures like flies that lay thousands of eggs, even if, that still means that a substantial number of my offspring are going to make it. But honeybees have merged these two systems together in a way that I think is quite remarkable. They have tons of offspring. The queen lays about 2,000 eggs per day. But where things really get interesting is that because her daughters are all caring for each one of those eggs and all of those larvae, they are able to have a survival rate of higher than 95% while also still producing tons and tons of offspring. It makes them incredible. It also makes them vulnerable because all of this brood, all of these baby bees are sitting inside of that comb undefended. And if you are able to get past all of the stinging adults, you've got an incredible little snack here. These little dumplings, which are full of fat and protein that's already been broken down. They digest their own bodies when they're transitioning from pupae into adults. So every time you see one of those jars at the grocery store that's shaped like a bear and it's full of honey, it should really be full of brood, because the bears are not particularly interested in the honey. That's not what they need when they're about to go into this period of lower metabolism. What they need is a lot of fat and protein. They're going after the brood. You probably wouldn't want a jar full of brood, would you? No? Not a thing. Okay, I understand. It's fine. So when we think about our bees, the huge question looming in everybody's minds is, what is going on with the bees? Why are they dying at these ridiculously high rates? And if I may, I believe it was H.L. Mencken that said, for every complex problem, there's an answer that is clear, simple, and wrong. And for many of us, those are the answers that we are seeking. We want a very, very, very clear, very simple answer. So when you listen, if you've listened to the media about this subject, you probably have an idea of exactly what's happening to the bees. Any guesses? What's killing the bees? Pesticides. I also heard pesticides. I heard pesticides again, and I heard climate. Yeah. So because of something that I would like to refer to as silent spring syndrome, we have focused so heavily on a chemical being at the root of every ecological problem that we tend to just focus on that when other things are coming to the surface. Over the past 30-ish years, we have been recognizing progressively that the pesticides are not the key feature that's actually killing the honeybees. Now, they are definitely a big part of the problem. We refer to the 3 Ps as the biggest issues impacting honeybees, and they are pesticides, pathogens, and parasites. But of the 3 Ps, parasites are the biggest concern. Now, that doesn't mean that pesticides are off the hook, but that means we need to consider what the parasites are doing in this system and give them more air than we've been giving, because at the moment, pesticides seem to take up all the air in the room. So I started focusing on these parasites and trying to understand, how is it that they can have such a dramatic, outsized impact on the bees? I learned very rapidly that honeybee parasites are a different sort of parasite altogether. They even have their own ecological category. They are referred to as melitophiles. Now, it's a portmanteau that you'll probably never see under any other circumstances except my talk, because when else are you going to hear this word? But it is a combination of 2 Greek words, melit, which is Greek for honey, and phile for lover of. So once again, we're back to this focus on the honey, but it's actually the brood that most of the melitophiles are after. And these creatures, they have had to refine so many aspects of their biology to actually go after bees, because bees themselves are so clever and so strange as superorganisms that they have created their own evolutionary category of parasites who have just really changed the game for parasitism. And the one that is of greatest concern to honeybees at this moment is right here before you. Many of you may not have even noticed, but you are currently staring into the face of evil. Not this one, don't look at me. No, right here. Right here. That is the face of evil. This is Varroa destructor. And while that name may sound a bit melodramatic, these organisms are anything but. Their ability to go into a colony and within a year cause the death of that colony is actually quite remarkable. It's also quite concerning that the bees themselves have very little effective resistance to this parasite. They're considered to be the agent of primary concern driving that trend of losses in the pollinator pandemic that I explained to you earlier. They are the key focus, but for a long time we didn't understand how they could possibly be the key focus. What is this tiny little thing doing, sucking out a small amount of the bee's blood and causing this many issues when blood is renewed every day in a bee's body? How can this thing be so problematic? This organism is one of the top 3 most economically damaging invasive species in the world. Think about that for a moment, because there are a lot of invasive species in the world, and this ranks as one of the top 3. They were first found in the United States in 1987. I can promise you I had nothing to do with it. I was -2 at the time. But they wiped out, within 10 years of their introduction, nearly every single unmanaged bee colony in this country. And it was quite remarkable. We have lost 99.98% of our unmanaged bee colonies. Now, something that really tipped me off, that these creatures couldn't be causing the damage that they're causing in the way that we think, was looking at their reproduction. So right there at the end of that paintbrush that I'm holding is the egg of a varroa mite. Now, it might not look that large to you, but by comparison to this flat little young lady that laid it, it's actually quite remarkable in size. Between 18 and 28% her body volume in that single egg, and they're able to produce one of those every 30 hours. They're pretty big eggs. They'll be like a human woman who weighs 100 pounds, producing a 28-pound baby every single day. Sorry to leave you with that very disturbing thought, but I just need you guys to understand why this looked so crazy to me the first time I saw one of these eggs, and it immediately struck me that there's no possible way that this organism could be feeding on something that is as nutritively poor as the bee's blood, if they're going to make something that requires this much of a nutritive investment. But as I looked through the literature, every publication, every poster, every fact sheet resounded with one voice: these parasites are feeding on the bee's blood. And so, if you've ever seen any of those shows, like, I don't know, Law and Order or something, where there's a journalist trying to find out some information, they're in the bottom of a library looking through microfiche, that was me as a graduate student trying to figure out how we got to this conclusion that this organism was feeding on the bee's blood. And the earliest research conducted about it was conducted in the former Soviet Union. And so it was very hard to find the papers. It took almost 4 months for me to actually get this single paper from a library in Russia, but eventually I was able to obtain it and found out, after having one of my colleagues translate it from Russian, that it actually never said that the parasites were feeding on the bee's blood. That was an issue with us having never really read the paper and only seeing the abstract. The abstract is a single paragraph summary of research that sometimes took 5 or more years, and it's summed up in that little paragraph. And in that paragraph, it seemed to suggest that they were feeding on the bee's blood, but it was actually a bit of a mistranslation of what it was saying. So my colleague, Eugene Ryabov, who is translating this for me, said, "Oh, Sammy, you might want to see this part. This is really interesting. It says that the parasite seems to feed on the bee's blood, but somebody should really test that." It's like, oh, well, that will be interesting as a PhD project. So I started focusing on this parasite, looking at this gigantic set of eggs. It's really what got me onto it, because that female that you see there is pretty flat. And when that egg is still inside of her body, it causes her height to almost triple. They're pretty large eggs. But then I had to take a moment and do something that I'm sure not a lot of you have had the opportunity to do. Like, what is it actually like to peer into an organism? I had some pretty cool special equipment in order to do it. What you see on my face there, those are actually oral surgery loops. One day I was getting my teeth cleaned, and the dentist peered into my mouth, and I said, "Oh my God, those are amazing." "What?" I'm like, those, can I get a pair of those? He's like, "These are for dentists." I'm like, "Yeah, but I think you might be thinking a little too small. Entomologists would love these. I can walk around with a microscope on my face." But it allowed me to get a really good look at these organisms and see what was happening under some circumstances inside of them. We were able to photochemically bleach some of the mites so that we could see directly through to their digestive system, and saw some really interesting things. The mass of food that was in their digestive system didn't resemble the bee's blood. The bee's blood is this yellowish fluid, and it's not particularly viscous. But instead, inside of the digestive system of the mites was this thick, viscous mass. And it was white. It was very interesting. So The question of what tissue are these mites consuming when they feed on honeybees was a big one that left me really interested in what was occurring under this set of circumstances. So one of the things that we were able to do was actually biochemically stain the internal organs of the bees. So what you're looking at right now, that is the abdomen of a honeybee. And this is an image after we have fed it two biochemically distinct biostains that would actually go to these different tissues, so it's tissue specific. We actually stained the bee's liver. So this red substance that you see here is the liver of the bee, referred to as the fat body, and then all of that yellow is the bee's blood. And so you might be a bit ahead of me at this point in guessing where we're going with this, considering that the bee's blood has very low nutritive content, but the bee's liver has quite a bit, high protein, high lipid content. This would be the tissue that I would be going after if I was going to have a baby that was 28% my body volume every day. So the expectation here is that if the mites are feeding on the bee's blood, then they will, their digestive system will show that bright yellow fluorescence. If the mites are feeding on the bee's liver, their digestive system will show that bright red fluorescence. And that's what we got all 64 times we did this experiment, which I thought pretty definitive, right? I think we have an idea of what's going on here. But if you're going to overturn dogma that has persisted for more than 70 years, more than twice the amount of time that, well, it's a very long time. And so I needed to figure out, how am I going to make sure that people can really see what's happening here? Well, I know that people really like to see these kinds of things with their own eyes, and so we took some honeybees, put them into the low-temperature scanning electron microscope, and actually used a feather blade to cut through the mite and the bee after we had frozen them solid. And we were able to see inside of both organisms, so you can actually see here. Actually, we're going to back up just a little bit because it took me a long time to get that image, and there weren't enough oohs and ahs when I unveiled it. So if we could, just for a moment, we're going to... Thank you. I appreciate that. I appreciate that very much. This was huge for me, actually getting to see this and notice that the tissue just on the other side of the membrane that the parasite is feeding through is all liver. Just a big old bundle of it. It even runs under this area as well. And it's a very important tissue to the bees. And so after looking through all of this, and continuing to do another set of experiments. I don't want to give you all the details in this presentation because it's not just about what I've done before. I want to talk to you about what I'm doing now as well. But this was really important for us to consider and did a lot to change the standing paradigm of what's going on with the bees. We have known that Varroa destructor is somewhere at the center of these issues. We've known for a long time that when colony collapse disorder was occurring, that these parasites were in some way involved. But we could not figure out what the link for all of this was. And now when you actually look at the sets of pathologies, there's been a constellation of 9 different pathologies that we have seen in honeybees that we have not been able to figure out what is the common link between all of them. And it turns out there are 9 essential functions of the bee's liver. And every single one of them, the disruption of it would cause the specific pathology that we've seen in this constellation of issues with the bees. So understanding how these parasites harm the bees has been paramount to us understanding how we can help them. But in addition to that, it may also allow us the information necessary for us to do something about these parasites. We're now working on a project in the Boulder Bee Lab where we are, well, the beekeepers at the American Bee Federation have referred to it as the Varroa birth control study, where we, having figured out that the proteins that are present in the bee's liver are what the mites are actually pulling out of the bees and using to construct their own egg yolk, we are figuring out how to disrupt that now so that we can stop these parasites from being able to effectively reproduce. And that has been a very exciting direction to go in with our research. See, these parasites, we thought we knew a fair bit about them. And this is actually a video captured by through some of the equipment in our lab, and it's showing you for the very first time what happens inside of a sealed brood cell. This cell is not open, but thanks to the system that we've been referring to as mite in sight, we've been able, I love puns, y'all. They're not going to stop, all right? we've been able to actually see what this parasite is doing to the bees. And there are a number of concerns there. I mean, you can see even in the brood that the fat body tissue is incredibly important. It's that ubiquitously distributed white mass just at the surface of that developing bee's skin. And we know these are all those pathologies that I was explaining earlier that have been showing up in the bees. But we also need to remember that if we take our eye off the ball and only focus on this parasite, it will be to our detriment. Because one of the reasons why we're in this position is because we waited for it to arrive before we studied it. And that's where this parasite comes in. That is the Tropolaelaps mite. It is a parasite that is at this time not found in the United States or the Western world. But this parasite is of huge concern. When it arrived in Pakistan in 1982, within only 8 years of it being there, it extirpated all of the honeybees within that country's border. We don't know pretty much anything about it. We have thought of it as somebody else's problem. This is a parasite that other people can deal with because it's not going to be a problem in the West. That is problematic thinking. There was a time where we even thought that about COVID. As a result of so little study being conducted about this parasite, open questions abound. We don't know what this thing is eating when it attaches itself to bees. That's the fundamental way that parasites impact their host. We don't know what happens when the varroa mite and the tropy mite are present inside of the same cells. And guys, I'm mostly just showing you this because I'm really proud of this system. I love that we were able to put this together and actually peer into these cells and see this parasite. This is the very first video ever captured of this parasite inside of a sealed brood cell. Yeah, sure. So an understanding of its life cycle is critical, but it's very hard to study its life cycle because it exists under the cell capping, conducting its life in obscurity and total darkness. So we are working to get all of this information available before this creature arrives. And trust me, with the route that it's currently taking around the world, it is on its way. So the Mite Insight system has been a great way for us to actually focus on this. You can see a picture that one of my students captured, one of the undergrads in our lab captured of this system. And it basically consists of an incubator box with a microscope over top of that, a slit cut into it so we can see inside, a camera, and a relay recorder that's actually able to record for up to 14 days, which gives us the entire 12-day period nonstop that the parasite is under the cell capping with that developing bee. Through this, we can learn exactly what it's doing, how long it spends in each segment of its life cycle, and potentially target any weak links in the system. Now, you may not have heard of Tropolaelaps mercedes-i, but it is a huge concern and definitely something that we should think of as an emerging threat to Western bees. But there's a reason why you probably haven't heard much about it. There was a paper published in the late 1980s that left us all with the impression that we didn't need to worry about these things. The title of the paper itself was Tropolaelaps clarii, which was the name of it at the time before we figured out that Tropolaelaps clarii was 2 species. So they were actually talking about Tropolaelaps mercedes-i. But Tropolaelaps, a serious pest of Apis mellifera in the tropics, but not dangerous for apiculture in temperate zones. So before this paper was published, there was a fair amount of concern and buzz where we were saying, oh, maybe we need to look into this. This could be the new varroa. And then this paper was published, and it was based on good data. It seems that this parasite is not capable of actually feeding on adult bees the way that the varroa mites can. And so it needs brood to be present in the colony for it to feed. And so if brood is not present, which is the case in winter, all winter long there's no brood present, this parasite could not possibly survive. So the logic here seems sound. But when you have not studied a creature enough to know what else is going on in its life cycle, sometimes important pieces of information that need to go into your logical chain are missing. One important piece of information that I don't think that people are looking at, and you are going to begin to see a pattern here. There's a paper written about this parasite in the Philippines and in China. The paper is written in Chinese and was never translated into English. So I had a colleague translate this paper into English, and is the very first paper to describe this parasite, and it calls it the Chinese rat mite. The parasite was found at the time only on rats. And I think that's a set of information that we have not been considering. the possibility that this organism actually has alternative hosts, and the capacity to persist, maybe even in rodent burrows, throughout the entirety of the winter. For 4 months out of the year, these parasites disappear from colonies in temperate regions, but they do exist in colonies in temperate regions. This is the current distribution map of Tropolelaps mites throughout the world at this time. And in 2020, when I first started tracking its spread around the world, this red area was the only region of the world where it was present. And that was already concerning, because before that, they were only present in Southeast Asia. So just this region. Then they moved into South Asia and East Asia, and moved into the coldest reaches of China and Korea, and we realized they are somehow able to survive the winter. They leave the colonies in temperate regions for about 4 months out of the year, December to close to the end of March. They're gone. We don't know where they go, we don't know what they do, we just know that they show up again in April. And this parasite is of huge concern because of the level of destruction that it's caused, but also the fact that it is the least species-specific of all of the honeybee parasites. There were reports of it on bumblebees in Thailand, and reports of it on carpenter bees in India. Carpenter bees are solitary bees. Its capacity to even go after them is of great concern. Also of great concern is the fact that just between 2020 and 2024, it spread between Uzbekistan, Kazakhstan, Tajikistan, Russia, and has recently arrived in Ukraine. Ukraine happens to be the primary region of the world where Canada gets its bees. So one of the primary regions of the world where Canada gets its bees. So this parasite could arrive on our doorstep anytime. So, you know, all those chants about building a wall aren't going to help there. So the work that we've been conducting in the Boulder Bee Lab has also taken the focus of figuring out if there's some way that we can control these parasites. Because they have seen so little attention, there is currently no pesticide or treatment method that is registered for the control of this parasite. So while in Southeast Asia, I and my postdoc, Madison Sankovitz, postdoc extraordinaire, we were able to test some elements of control and even methods of monitoring against these mites so that we could figure out how do we figure out where they are and whether they're in a colony that we're sampling in. So Madison and I have been working on that together. But even before I had a postdoc start in my lab, I was working on figuring out what can we use to kill them. And formic acid seems to be a pretty effective measure against them. We submitted a paper recently. You can actually look at the preprint now, but hopefully will be published in the next few weeks. There's no telling with papers anymore, but hopefully it'll be published soon. And you'll be able to see the efficacy of this set of chemicals, which is, we are currently working to get this approved for usage against this parasite, but we also tested non-chemical treatment measures using these thermal heaters to actually heat the colony and see if heating the parasites would actually lead to their demise. It could, but unfortunately it was a bit too variable, and the formic acid, which we soaked pieces of wood in formic acid and pushed them into the front of the colonies, and then also compared a method that uses the packing material that's under hamburgers. You guys, like that very absorbent stuff, soaked in formic acid. Both of them were about as effective at killing these parasites, but they were able to kill more than 90% of the parasites in a colony. So we do finally have a single effective treatment measure to be used against this parasite, which I'm very proud to report. The work, that whole set of work, has led me to a very specific conclusion. Our Western-centric focus on honeybee health is to our detriment. We have one species of honeybee in the Western world. One. Every other honeybee species that exists in Southeast Asia. That is the only place in the world that you can go and find every single species of honeybee. And it's quite remarkable that that is also one of the regions of the world where the least honeybee research is actually being conducted. A lot of that is because of the vestiges of colonialism, and I won't get into all of those details. Don't want to ruffle too many feathers in my first talk here, but these are things that we really need to consider. So I started a honeybee parasitological survey, where it was my goal to go to a few different countries, collect parasites from those areas, analyze them, sequence their genome, better understand them. It was a pretty ambitious project, and realized that it was going to be very difficult for me to actually conduct this. But the more that I worked on this survey, even with, at the time, less ambitious goals, the more others joined in. So the United States Department of Agriculture has become a partner in this project. Then National Geographic became a partner in this project, and it has become a lot more feasible for us to go to every country where these honeybees are, to sample them, and to look at all of the different parasites and diseases present therein. So for that project, we have even opened our plans to doing something called the Honey Genome Project. So this project, a pretty exciting one, we are collecting all of these species, and we've decided to actually sequence the genome of every single honeybee species, make that available, of course, open access, which will teach us a lot about the bees, because something that's really interesting is that Apis mellifera is the only honeybee species that left Southeast Asia, moved into the Western world, moved into Europe, and it was, in some ways, a pretty great idea. This bee had the ability to escape all the parasites that were swirling around this area where there were tons and tons and tons of species of honeybees, and moved into a place where it was the only one. It left its parasites behind, it left the competition behind, and it lost, at the same time, its parasite suppression genes, we're hypothesizing. And so looking at what genes allow all of these other species to be so competent at controlling parasites will be really helpful for us, because it is very pronounced that if you expose any of the other bee species to their parasites, they're really great at getting rid of them. But if you expose Apis mellifera to them, they seem to be unaware that they even have a parasite on them, and it is really sad to watch. And we have to understand why it is that that occurs if we're going to help our bees over in the Western world. We're also trying to develop a system, so the United States has contracted us to develop a system where we can actually determine whether trophy mites are present in a colony. So the only system right now is called the bump method. You take a pan, you grab a frame of brood, and you slam it against the pan, and hope that it dislodges the mites therein. Only problem is that it kills all the brood on that frame. And so beekeepers have been less than enthused about investing in this method, surprisingly. So we've decided that instead of just developing a system for determining whether these parasites are present that will be kind of expensive or time-consuming, we also want to develop something that can be employed easily in the field that's a low-tech method to use here. So we were asked to develop one. We're interested in developing two different systems. Here you can see that we are collecting some detritus from the frame, and we want a system where we can actually swab it the same way that you can stick a swab up your nose and use a lateral flow test to determine if you have COVID or potentially other diseases in the future. We are hoping to develop something similar to that for these mites by just swabbing the bottoms of the colonies, the tops of the frames, and seeing what eDNA is left behind by these creatures and what we can do with that. And you can see here that's Madison, my postdoc, actually smoking the frames, and then we apply a transparency to that frame. Does anybody remember transparencies? Like pre-PowerPoint? Anybody? So there are some people who are old like me here. Okay. So the great thing about these transparencies is that you can apply a layer of glue to them, press them against the frame, and then pull off whatever may be sticking to the frame. So we've been doing this in the wild, running around Asia, trying to determine if this will be an effective measure, a low-tech method of just determining whether these mites are even present in a colony, which will be very important for developing an emergency response plan in any countries where these parasites arise. And this is the detritus that we found at the bottom of a colony. There's a spider running around in it for some reason. And we are hoping to actually swab that and really create a system where we can see even more accurately whether these parasites are present. We are also working on something called the Vespa Tracking Initiative. You may have heard about the hornets that arrived in the Pacific Northwest that consume honeybees and were affectionately referred to as the murder hornets. You can actually see one right here attempting to murder me. She is failing, but I think ever so often they want to know if they still got it, you know? So we have been collecting hornets from all over Asia because we need to determine what exactly has been the donor population for the hornets that exist in the Pacific Northwest. We had more than one introduction of hornets, at least 3 introductions of hornets in a very small amount of time, and potentially from different regions. And what we need to figure out is where are they coming from, and how can we stop these systems that are allowing them to show up so readily. Madison has actually been taking the lead on that project, and Madison is one of the first people, actually the first person that I hired at the Boulder Bee Lab, and it was a great decision. She has been incredible, huge amount of help, and somebody that seems to be able to travel anywhere, get a little bit of jet lag, and be totally fine. I'm slogging around like, oh my gosh. She's like, oh, ready to go. So that's been awesome. And these are the countries that we've already visited: Thailand, Korea, Japan, and Bangladesh. Bangladesh being the most recent. But we still have to go to Mongolia, Sri Lanka, either Nepal or Bhutan, Java, Sumatra or Sulawesi, and the Philippines. So we've got our work cut out for us. But when I tell you that this experience has been so rewarding that it doesn't actually seem to be the slog that it may seem on this slide, well, let me show you a little bit of what we've been up to. How does the volume work up here? Are we good on the volume? Yeah? All right. I'm going to fire this up. Let's see if you guys can hear anything. Nope. We're just going to press that media button, and we're going to try it again. Oh, I didn't realize I froze it on my face like that. That was great. Today, we have a pretty epic journey planned. Our goal is to find as many colonies in this forest over the next 3 days as we possibly can. And we're really focusing on a tiny little parasite called the trophy mite. Pretty cool, right? So you might be wondering, oh, thank you. Thank you very much. You might be wondering, hey, what you doing there, buddy? It's like a whole little production there. Part of what we do, I mean, I know the name of my lab is the Boulder Bee Lab, but one of the reasons why I was hired here is because I'm a science communicator. I love to communicate science, and we are finding more and more effective measures to do this. And one of those ways is to actually bring people with us on these trips. And so we have a media team that comes with us, National Geographic photographers who are there with us, as well as a videographer. And we talk through everything that's going on as we're conducting this work. And this was day 1 of us being on a boat for a very long time. What was it, 8 days on a boat, sleeping on the boat, living on the boat, all together. It was, I'll tell you, when they first told me that in order to get through the Sundarbans effectively, you have to do so every day on a boat, I was like, well, that's the end of this project for us then. Bye, guys. I hated the idea. But the things that I will do for bees, I tell you, I thought about it. Madison gave me a little pep talk and was like, we can do this, man. We can do this. And we lived on a boat for 8 days, and it was kind of incredible. We got to drift through the forest. We were able to stop at a number of different locations as well when we were in Bangladesh, where we actually interacted with the beekeepers there. And we realized pretty quickly that it was very important for us to have an exchange going on here, where it's not just us showing up and extracting information, but us making sure that we are leaving the populace there with all the information that they need to recognize the parasites that are present there. And so, we noticed when we showed up with all of our equipment, the beekeepers, they just grabbed their phones immediately and were like, what's happening here? Tell us everything. And with our translator, we walked them through the biology of the trophy mites and all the problems that they cause. And it was pretty incredible. You can see that entire villages gathered around. Every location that we went to, the whole village showed up. At first, I wasn't really sure what was going on. I showed one of the kids one of the parasites, and she ran away. But then she came back with droves of people. And we realized, okay, so she wasn't so afraid of the trophy mite. She was enthused and wanted other people to see what was happening here. Because here's the thing. Under most circumstances, they're not going to get the opportunity to see a parasite that's 0.5 millimeter in width. It's very difficult to actually locate these and see these inside of the colonies. And so it's been really, really, really helpful for them to be able to look through Shin, our photographer's viewfinder, and see these incredible parasites, to see images like this. That is my finger, by the way, right next to it, so just so that you can have an image of scale. And I'm holding a paintbrush with the other hand. And at the same time, we're doing the research. We're putting those parasites into the actual tubes that we're using there. And then after that, we do something that everybody loves. We drop them into liquid nitrogen, which is negative 196 degrees Celsius. And when I tell you, kids' faces light up for liquid nitrogen, I mean it. And honestly, I mean, I bet your faces would light up for some liquid nitrogen, right? I mean, it's pretty incredible stuff. Let's see if you can resist responding the way that they do, because check this stuff out. Pretty cool, right? Yeah. Still got the chill from that shock wave all the way down there. So when you are taking out a giant canister of liquid nitrogen and dunking parasites in it, people get really interested. And this allows you an incredible teaching opportunity where you can talk to them about these parasites, teach them different elements of bee health and bee disease diagnostics. What we didn't quite know was going to happen on this trip was that the beekeepers were going to be teaching us so much. There were several colonies where we could not find a single trophy mite. Some apiaries where we couldn't find a single trophy mite. And when we asked the beekeepers about this, they were quick to tell us that the plants that are near these beetle leaves, they were saying, and we never really fully got the exact translation of that, but we're working on figuring it out. They seem to do really well in the face of these parasites, and the parasites don't stick around for very long. We opened several colonies where the leaves were crushed up in a little basket on top of the frames, and we wanted to better understand them. We also did a little bit of comedy along the way, just impromptu. This is Chris, our apiary manager, acting as if he is eating the larvae, and that seems to be quite to the delight of the kids there. And then, confusingly enough, a few of the women in the village decided that they apparently wanted Madison to adopt their child. So they just gave their babies to Madison and then took pictures of her holding their children. I don't fully understand what was going on, but I feel like we were accepted, and I loved it. This gentleman even proposed to her. This was her response when she said no, and I'm glad that he took it so well. And then all of them gathered around Rico, our videographer, because they wanted to see what he was looking at. Rico's task was to get a bird's eye view of all of these locations, to get an idea of all the plants that were around, all the agriculture in the region. And so he's flying a drone over and taking video and picture that's geotagged so that we'll be able to compare that to the data later and better understand what areas these trophy mites are present in and what's nearby those colonies. We also learned very rapidly that one of the great ways of outreach is to just give a kid a bee. So you might look at this and think, oh, that's risky. That bee could sting that kid. That kid might be allergic. What are you guys doing? You're going to get the BioFrontiers Institute shut down. No, calm down. Yeah, okay. Well, one of the great things about this is that with these bees, we are possessed of some interesting knowledge. The first day of life for honeybees, they are incapable of stinging or flying. Their exoskeleton has not fully, the proteins in their exoskeleton have not fully cross-linked, and so they're not solid enough to stick a stinger in you or fly away. So they make great pets for 24 to 36 hours, and they'll just crawl around every surface, they'll lick you, they'll drum on you with their antenna, and that allowed us to give these kids the opportunity to really interact with these bees in a way they thought they never could. While we also had our translator Mowgli explain to them, you can only do this on day one, kiddos. So it was quite the experience, to the point where there were kids that didn't want to leave by the time we finished this work and had to get going. You can also see that I did not have access to a razor at any point in that trip. So yeah, that was a thing. But we learned a lot from the beekeepers there, and it was an incredibly exciting experience. I mean, just when we left that apiary, we were so excited. Look at the excitement on our faces. We could not contain it. It was such an incredible experience. I don't have time to actually walk you through all the stuff that happened, but here's the super fast version. We were on a boat for a long time. We contracted some individuals to help us from a local village. They were able to create this 3D map of the forest in their minds and find all the honeybee colonies, while we attempted to do the same and failed miserably. They even helped us find hornet nests. You can see Madison and I looking through this nest of hornets, and we found several colonies of Apis dorsata from which we collected the adult bees. This is the beekeepers creating their smokers, which are just a bundle of reeds that they are actually using there to smoke away the bees, which was incredible because throughout this entire trip, they never got a single sting. And this is me asking them how that is even possible. And it was quite the experience, one that we will never forget. We were even able to find a species of varroa mite that we've been looking for a long time that is the most closely related to the one that we have in the US. We really wanted to look at its genetics and better understand how it differs from other varroa mite species. We spent a lot of time looking for them, eventually found them, and we're so excited to get the opportunity to do this kind of work. And one of the reasons why we can do this kind of incredibly ambitious work is because we work so closely with, because my lab, the Boulder Bee Lab, is a part of the BioFrontiers Institute, and I'm so grateful that the BioFrontiers Institute has invested in my lab, but also provided all the support and let me have these crazy, ridiculous trips where I'm darting over to the other side of the world. I'm really working in a context where the individuals that I'm working with have been incredibly supportive of the odd work that we're doing. The Boulder Bee Lab doesn't just work on one thing. We focus on symbiotic relationships, superorganism biology, insect behavior, and my graduate students are just as diverse as those subjects. We have students studying stingless bees in Peru. My PhD student, Kat, is working with a National Geographic explorer in Peru who is considered to be one of the 50 people changing the world that you should know about. And this interaction between the two of them, I'm expecting to be a quite remarkable set of work that's just getting off the ground this summer. My graduate student, Lincoln, also one of the PhD students in this lab, is doing some incredible work to see what impact the varroa mites have indirectly on the native bees as they transmit viruses to the honeybees that the honeybees leave behind on flowers that those native bees can then pick up when they visit the same flower. So what indirect impact is varroa having, even though it doesn't feed on native bees? What is it doing to them? And we even have undergraduates conducting remarkable research, as Justin Hine has been working very closely to help us track the set of proteins that the varroa mites extract from the bees into their eggs, which you can actually see here in these micro-CT images. And it has been quite an interesting adventure. I am so grateful for everyone who contributed to this project, both in the United States, as well as our international collaborators, as well as our funding through Project APIS-M, Manuka Doctor, the USDA, National Geographic, HiveFlow, and I would like to thank all of you for listening. And if there is any time for question and answer, I would love to answer whatever questions you have. Looks like we got one over here.
When you were talking about the formic acid as a possible treatment, were there any negative effects on the bees?
Oh, yes. Great question. And if I had time to actually talk about that whole project as its own presentation, I would have gotten into this. But formic acid is incredibly volatile, and it is even more so when it is very, very hot. And so anything above 84 degrees, and that formic acid volatilizes in such a way where it can be damaging to the adult bees as well as to the tiny little mites themselves. So you're trying to kill a bug on a bug. You need to get exactly the right concentration in set of circumstances. Here in the US, where we have temperate environments in some parts, we can more effectively utilize that. But places like the South, Florida, Texas, will have limited utility from this specific chemical, which is why Madison will actually be heading to Bangladesh to test 3 more treatment options in about a month.
Sammy, your list of Asian countries that you're going to visit, you don't have Vietnam,
Correct.
you don't have Laos, and you don't have Cambodia on there. And I would put those 3 countries on your list. The women up country in Vietnam chewed a nut called the betel nut.
Yep.
Turned their teeth black.
Black. I've seen it. It's
Black,
fascinating.
black. And you probably find the tree there.
Yeah. So Vietnam, Laos, and Cambodia would be great regions for us to conduct research about honeybees, and originally all three were on our list. We have had some issues, all of which I will not rattle off while on camera, but just to say, it can be very difficult to conduct research in certain parts of the world and do so in a way that is above board. And because we were asked for bribes very openly, we've decided that to ensure that we are doing things by the book under all sets of circumstances, we do not do an ends justify the means system of, all right, we'll bribe who we have to bribe to do the research. There will be none of that. Hey there.
In a colony, are there just female bees or are there also male bees?
Oh, that's a great question. Okay, so in a colony of honeybees, things get pretty wild because the system of generating offspring is a bit different from how it works in humans. When a queen honeybee goes off on a mating flight, that's the only time that she ever leaves the colony. She comes back and she has actually stored the genetic material of all the mates that, or individuals that she mated with inside of a special organ where she can keep them alive for years, more than a decade, actually feeding the sperm of those males. Now, then where things get interesting is that she has the capacity to choose the gender of her offspring. She can choose through muscular contractions to allow for an egg that's passing through the canal to be fertilized or unfertilized. If it's unfertilized, it'll be a boy. If it's fertilized, it'll be a girl. Imagine if we lived in a world where people could make those kinds of choices. This would be a ridiculous place to exist in. But this is just a bee thing, and some ants, and some wasps. So the fascinating thing about this is that the colony is primarily composed of females, and you don't want that to get out of balance because the ladies are the workers in the colony. They get it done. The men, they just walk around and they eat. And they are so inept at what they do that they cannot even feed themselves. Their shoulders are too big for them to get into little honey pots and feed. So they have to have their sisters reach their tongues in there, extract the food, and regurgitate it into their mouths. They are not always great to have around. And so at the end of the year, when they need to conserve energy, they kick all the guys out of the colony, and they live the rest of their lives with just the ladies there until the spring begins again, and they will have some more dudes in the colony. Mm-hmm. Yeah.
Dr. Ramsey, I wanted you to clarify for me, when you showed the pictures of the tropy mites, I thought the tropy mites were reddish orange.
Oh.
Did you show a tropy mite that was transparent, or was that just the camera?
That was just the camera.
Okay.
So the way that, when Shin was trying to light these tropy mites, our Nat Geo photographer, Shin Arunrugstashai, is responsible for most of the pictures that you saw in this presentation. When Shin is trying to light them, he has a very hard time doing so. They're so small, and he has to use a macro lens, which cuts out a lot of the light. So he uses a really special light on two sides of the insect, and because the light is so bright and their integument is so thin, it can make them look translucent, but they are a sort of tan, almost reddish color. Yes.
Hi.
Hey.
When you were discussing trying to determine whether a colony is having an issue with these mice, my first thought, because you kind of tied it into COVID at the beginning, was grind up some of them and PCR amplify the genes for the mites. Then you immediately said that you want it to be like easy to use in the field, and I was like, okay. But has anyone thought of like a rapid test sort of equivalent where you're looking for the proteins in the mites and also grinding up some of the baby bees?
So that's the goal. But it's going to be a little bit more difficult because we've got to wade through so much junk in the colony. The detritus at the bottom of the colony would be the fastest and easiest way to do this. So stick a swab into all of the stuff that falls out of the colony. There's this rain every day. As the bees are walking through the colony, they're cleaning out cells, there's bits of legs from parasites. If we can swab that content, you won't even have to disturb the colony itself. You can do that without even opening the colony. So that's the goal. But we may find that there's just too much noise in that system, and we may even have to go a bit more invasive. We may even have to grind a few things up, which I would hope not, but we'll see. Yeah.
Yeah, could you say more about the, I think, beetle leaves
Yeah.
that you learned from the beekeepers?
Yes. So I can say more about it, but I wish that I can say more than what I can say, because there's not a lot that we know about them. We just know that there are these plants that come, or there are these leaves that come from a tree in that area. We took some pictures. We have yet to fully identify it, but we have determined it's not the same plant that the beetle nut comes from. So it looks like they're using the same name for two different plants, which happens frequently when you're dealing with translations there. We know of a lot of plants that have volatiles and chemicals inside of them that are capable of warding off insects. They have a repellent effect or poisoning them. Things like nicotine. Nicotine is a poison, everybody. The plants are employing it as an insecticide, and we are using it for fun. Weird. But there are plenty of chemicals of that nature that are naturally produced by these plants. So it wouldn't actually be shocking if it actually has the impact that they say that it does. What does tend to happen under these circumstances, though, is that people ignore that kind of information because it's coming from a non-peer-reviewed resource, it's coming from indigenous tribes of people, and I really would like for us to begin to listen to individuals who have spent so much time in these circumstances, really in the trenches with parasites that we know almost nothing about.
Over here, Sammy.
What happens when the queen bee dies? Who takes over?
I love these questions. We got to keep nurturing this curiosity, because something that I will tell you is that, like, the more that I do outreach, the older that I go with the kids, like, the less I get exciting questions like that. And I think it's something that we do sometimes in education, where, like, something that we do seems to squeeze out a lot of their curiosity. So I love how curious you all are. Honeybees, they have a really interesting system going. It's an all-for-one, one-for-all kind of thing. Everybody needs to be protecting that queen, because if she dies, the entire colony can die. The only bee that is capable of producing life in that colony is the queen. Everybody else takes care of the life that she has produced. So if something bad happens to her, you can lose that colony. Now, if the queen dies unexpectedly and there's still brood in the colony, they can create something called an emergency cell, where they will switch the bees that have been fed in that cell to royal jelly, and if they feed them enough royal jelly, they'll develop into a queen. Now, every honeybee baby gets royal jelly at some point in their lives, usually for just 3 days. And then after that, they get switched to a set of food that's just composed of honey and pollen and other stuff that the bees regurgitate to them, and it kind of starves them in such a way that the fullness of their anatomy doesn't develop. But if you feed them royal jelly every day of their lives, their ovaries and all their reproductive system will develop in such a way that they can become a queen. If there are no bees that are young enough in the colony to become a new queen, that is the end of that colony. So it is really important to protect the queen, and that is one of the reasons why those bees have a stinger on their back end, to make sure that they can very effectively say, hey, back up. You are making poor life decisions getting near my queen, all right?
Okay, I'm going to ask the last question.
All right.
So we've got all these other honeybees in Southeast Asia.
Mm-hmm.
They're resistant to parasites.
Yeah.
Why don't we bring them over here to pollinate our crops?
Love it. Love it. This is what I mean about those questions. That seems like the best answer to this system. If Apis mellifera is dying all day long from these parasites, has basically no parasite suppression ability, why don't we just let them go and bring over the Apis dorsata bees that you saw earlier in that tree? They're incredible at controlling parasites, and they are ginormous, almost twice the size of a regular honeybee. So, you know, that could work out great. Or would it? So, Apis mellifera made an interesting bargain evolutionarily. In losing their parasite suppression genes, in losing a lot of the energy that would be dedicated to dealing with competition and dealing with parasites, instead they dedicated that energy to producing huge colonies. And to produce huge colonies, they also need a lot of food. So they produce more than an order of magnitude greater an amount of honey than any of the other honeybee species out there. I mean, huge amounts. You can get 200 to 300 pounds of honey from a honeybee colony in a single season. And from their nearest relative, the Apis cerana bees, you can get between 4 and 12 pounds. You know, so that's a big difference for the honey producers out there. But in addition to that, the size of their colonies is really important. For pollination services, having a workforce that's 60,000 individuals strong allows them to pollinate so many crops that we can have them be what underpins our global food security. We can produce ridiculous amounts of food on a very small amount of land. They allow us to be sustainable in maintaining the population of people that we are maintaining. But if you go with one of the other species, many of them produce colonies that have between 500 and 2,000 individuals, and you will lose a remarkable amount of efficiency if you decide to switch to those other species. They also can't survive outside of a tropical environment, so that's also a big deal.
Yeah, but with climate change, we're all going to be tropical.
Oh, Roy!
Anyway, okay, so, thank you.
Thank you.
Let's thank Sammy once again.
Thank you. Thank you very much.